Good day, everyone. Since I have very little time, I will try to be brief and I will try to get to the point and present my case that this small portion of the repeat analysis topic is very suitable, is simply perfect as an introductory chapter on statistics and machine learning and bioinformatics. But still, a few words about the problem itself. So our DNA contains the so-called mobile elements, which are sequences that can move around the DNA. That some of them operate in cut and paste mode, so they cut themselves out and paste themselves somewhere else. Some of them are in copy and paste mode, and mostly we are here interested in the so-called lines and signs, long and short interspersed elements, which are basically genome strings that have been copied a lot over the course of the evolution. So, for example, the main example of such elements is the family of alluripids, which is basically a string of about 300 nucleotides that has been copied over 1 million times in the human genome. So if you do the math, it's about 10% of the entire length of the genome. Just copies of one string with, of course, they're not exact copies, they're copies that have undergone a lot of mutations along the way. So basically from the computer science perspective, the life of mobile element is quite simple. It is quite uneventful for a while. It just undergoes random mutations, and then at some point it can become active. Of course, not all of them become active, but some of them become active, and they copy themselves maybe several times around the genome, and then they go to sleep again and probably never wake up. So the problem is that after this long evolutionary process, what we get, for example, if we take a human genome and we take a single genome and we take, what we actually get is a snapshot of the current situation in this evolutionary process. So if we study the copies of this mobile element, we can first identify the subfamilies that are closer together and that have probably originated from a single source element maybe not so long ago. And then we can try to reconstruct the phylogenetic tree of how these repeats came around. And here lies my first point, which is that this is actually a very compelling problem. I mean, when you go into bioinformatics, if you go into bioinformatics, you're probably interested somehow or the other in evolution, because this is the main underlying theme of it. And here we are directly reconstructing the evolution. So it may be just direct phylogeny, like there have been studies that that constructed phylogenetic trees of primates based on alluripids, or it can be population genomics and maybe some analysis inside a single species, but still it is basically reconstructing the evolution, which makes for a very easy to understand and very compelling biological problem. Now, I'm done with biology at this point, so let me get to formalization. And again, I will only be outlining some main themes of this proposed chapter, and I will try to convince you that this is a good way to introduce many machine learning concepts. So basically, the life of a mobile element is very easy to formalize as a random process, and it is a very natural formalization. So each element is a string of 4 letters over the alphabet of 4 letters, and we begin with some original set of source elements. There probably were several of them, but it doesn't matter right now. And basically, we can view it as a continuous discrete process, as a continuous or as a discrete process, but still, at every time step, there is a small probability for a letter to be mutated, and at every time step, there is an even smaller probability for a whole copy to become active and copy itself a few times, right? So this formalization is very simple. I have explained it, and I think all of you can immediately, all of you can see that this is a more or less complete formalization, but I haven't introduced a single formula so far, right? But what is most important here is that it is a quite accurate model. So it is often the case that there is some underlying beautiful, very simple and elegant model, which then doesn't work in real life. For example, yep.
So you think that the bit there's no insertion deletions?
Yes, of course it is simplified. Yes, of course. So I know if there was a deletion, you can artificially insert a letter there and say that it was mutated, something like this. But still, it is actually a quite accurate model, which is very seldom the case, which is not very often the case in real life. For example, I don't know, in genome assembly, you have this beautiful, beautiful idea of the De Bruijn graphs, which are very helpful for genome assembly, and all assemblers are based on them. But if you take the original idea of constructing the De Bruijn graph and then going around in a layering cycle, it simply doesn't work. And of course, assemblers don't do that. Well, here is a simple formalization which does work. Yeah?
So the location of the house not getting to the model?
I think not, no. I will assume that we have found all of them. And I will not speak about the...
So you just have a bunch of sequences?
Yes. Yes. The locations are outside of the model, yes. Right. And this simple model also suggests that the problem immediately and naturally splits into two problems. First, in this set of sequences, we try to identify subfamilies. Some of them are subfamilies of larger subfamilies, etc. And then, already having identified the subfamilies, we reconstruct their phylogenetic tree. So my second bullet point here is that this problem admits a simple but accurate formalization, which leads to a natural decomposition of the problem. Which is, well, it is a, yeah, let's say that this is the set of elements that have originated from a single source. The set of elements that have originated. No, this is the problem. I mean, the problem is to find the subfamilies and reconstruct the tree. Yes. Yeah, it's not immediately obvious on this simple example, but even if you know the subfamilies, you still have to order them in a tree. You still have to find the structure. But this is simple, yes. It is simple, and I will speak about it in a minute. All right. So let's get to that. So now that I have presented the problem, let me say what are the methods of solving it, right? And I would propose to begin with the second problem, which is simpler, and which is, suppose that we have already identified the subfamilies. How do we reconstruct the tree structure? How do we know which subfamilies come from which parents, right? And there are basically two very simple ideas here. The first very simple idea is that, well, if a subfamily is older, then its elements have had more time to randomly mutate. So we can expect that in an older subfamily, its variance, like the radius of this subfamily, will be larger. So we cannot make any statements about the actual age in millions of years or something, because evolution is the rate of evolution changes all the time. But we can actually estimate we can actually infer some kind of an ordering. So we can basically look at two subfamilies and say that we are nearly certain that this one is older than that one. And once we have this ordering, then it is a simple application of the Bayes theorem, which is that for every subfamily, for every cluster C, we simply find the maximum a posteriori hypothesis. We find the subfamily which has the maximal, which is the most probable ancestor of this cluster among the subfamilies which are older, right? And again, there is a recurrent theme here in that it is a very simple and direct application of the most of the underlying principle of all machine learning, and there is basically virtually no complication here, but it does solve the problem. I mean, the problem is quite simple, so this direct application of Bayes' theorem is actually a valid approach to it, right? So I think this is also very good from the educational point of view in that we get a real-life problem that grows upon the very basics of machine learning and that does not require any complicated methods and lets us gently introduce the Bayes' theorem. This is my third point. And now we move on to the more complicated part, which is, of course, subfamily identification, that is, clustering. And here again, even though it is a more complicated problem indeed, lots can be done with relatively simple methods, right? But first, I would begin with a demonstration that if you blindly apply a clustering approach that looks natural, which is just take a bunch of strings, compute the Hamming distances between them, and try to cluster in this space, this will not work because it will fail to detect correlations between mutations, and subfamilies that are very clear will become very confused in the Hamming in this space. So this is also a good educational point, but then we can move to a very simple idea that lets us introduce statistical testing, statistical hypothesis testing. And this is the idea that correlations can be found by collecting and computing the so-called biprofiles. A biprofile is a very simple idea. Suppose you want to test whether two mutations are correlated. How do you do that? You compute the number of their co-occurrences, you compute the number of occurrences of each individual mutation, and then you compute the p-value of this number of co-occurrences under the null hypothesis that they are independent. So again, this is like the most basic application of statistical testing you can imagine, but it is a valid way to tackle our problem, and while it is probably not the best way, it still identifies reasonable subfamilies. So point number 4 is that biprofiles yield a simple introduction to statistical hypothesis testing. But this non-parametric technique did not use our probabilistic model, our generative model for these subfamilies. So maybe we can use it and get better results. And indeed we can. But here again, I would begin with an algorithm which is good, but which we will not ultimately use. And this is the expectation maximization algorithm, which is again, which is a very natural fit in this case, because we have a clearly defined generative model. It has latent variables which represent the subfamily assignments, and we can use the EM scheme to actually find local maxima in the likelihood in our model. But of course, EM is very slow, and for allo repeats it may be prohibitively slow. That's one point, and the other point is that there is a complication with number of clusters, because regular like the Bayesian information criterion does not really work here because we have additional structure in the clusters. So, but I do not propose to go into these problems in the chapter. What I do propose is to introduce the EM algorithm with a very simple example. But the good stuff here is that it is not a mixture of Gaussians. Because in nearly every book that introduces the EM algorithm, you get an example of fitting the parameters of the mixture of a few normal distributions. And here, it's still clustering, still the same stuff, but at least the distribution is different. And so with that, I would move on to the final part, which would be devoted to RETS, which stands for randomized test and split. And this is basically a randomized clustering algorithm based on the same idea of biprofiles. But now it gets a little bit complicated. So the algorithm goes as follows. First, choose a number of random pairs of mutations for testing. Then test them for independence. I'll get to that. If we find pairs that are not independent, that are correlated, this is a direct indication of how to split the subfamily. So if there is a correlated mutation, we take out the elements that contain this correlated pair and generate a new subfamily out of them. But of course, we would get lots of spurious splits, and we might get reasonable subfamilies split into several parts. So after splitting, we try to join subfamilies back. So we basically unite them and again test if we can break them apart. So that's the essence of RETS. And from the educational point of view, RETS lets us present a lot of interesting stuff. Well, first, the independence test is based on the notion of mutual information between pairs of positions, and the theorem that if the positions were uncorrelated, it would follow the gamma distribution. So this would lead to a statistical test for independence, right? And the second point here is, again, another application of statistical testing is that we need to somehow estimate the number of pairs that we need to test in order to have a reasonable chance of, in order to have a very small chance of missing a correlated pair. But there is also a larger theme here, which is that even if you do have an off-the-shelf algorithm which is good for this problem, which produces reasonable results like expectation maximization, still in bioinformatics, such algorithms don't always work because data sets are massive, and even if the algorithm is correct, unlike distance-based clustering from a while ago, still maybe you need to find a simpler approach, a faster approach that will work with this on this scale, right? So the sixth point is RETS, which is a more complicated but faster algorithm, and it shows some more in-depth statistical ideas. And my final point is that, of course, bioinformatics being a very practical field, would need a lot of exercises. And here you probably can already see that the proposed chapter has a lot of opportunities for exercises. Because first, there can be lots of mathematical exercises in elementary statistics, elementary probability theory. And second, basically, every single algorithm that I have briefly described in a hand-wavy way is actually very simple, and it can be made into a software engineering assignment for a student. So again, because the topic is rather simple, it provides many accessible exercises in both mathematics and programming. And this almost concludes my talk. I just want to review again the chapter plan, which would begin from the introduction about the mobile elements, then the evolution model and the formal problem setting, then reconstructive phylogeny, where we introduce the main topics of Bayesian inference. Then we move on to identifying subfamilies and speak about statistical testing and failures of distance-based clustering. And then we move on to model-based approaches, introduce expectation maximization scheme, and finally RETS. And here are my points in favor of this topic being a perfect match for an introductory chapter. And with that, I probably conclude my talk. Thank you.
Okay.
phylogeny construction. Just construct a subfamily, find the parent in this subfamily, and then construct subfamilies on the parents. And this kind of hierarchical, iterative application of your subfamily identification.
But that is exactly what is being done here, I think. Yes, actually, yes, you can construct a unified model that would unite both subfamily identification and phylogeny reconstruction. But this problem is so simple that we do not need this. We would certainly need it if we had missing links in the tree. For example, if we only had like we have in other phylogenetic problems, we only have the leaves of the tree, but we do not know the internal structure, and we do not even know the internal nodes. In this case, we need structural expectation maximization, which is definitely not a topic for an introductory chapter. Okay, but in this case, since we assume that all elements are preserved till the end, and we have all the nodes of the tree, you can split the problem in two. And this is, yes.
I want to reject the paper. That claim that the repeat finder constructed by the authors is better than other repeat finders because it finds a tenfold more repeats. The point here is that unlike gene or transmembrane segment or binding site or transcription factor, repeat is something with very vague biological definition. I mean, all definitions that we introduce are technical, but more or less by necessity.
Right.
And the question is, well, question slash suggestion is that you should deal with that somehow in the introduction of
I agree.
Chapter, because I mean, it's really an issue and it's not appreciated by many people. And the other point also that also might be relevant is that, for instance, in bacteria, a very frequent situation is that you have sort of a family of repeats, but none of them is full-size package. They overlap. You can't exactly say where the boundary is, and you have this fuzzy set of overlapping fragments of overlapping regions. You want to know what I mean about that if you work with.
Yeah, that's true. And well, of course, I should mention this in the introduction, but of course, I do not want to actually introduce these complications in the model. I want to keep it simple, and I want to assume basically that some kind of repeat finder has been run, and it has produced more or less well-aligned set of repeats. Because if that is not the case, then the models become much more complicated, and it's not an introductory chapter anymore. Yes.
So I have two comments. They are both related to what is best to teach first to newcomers to this from the biology side. One is that the clustering solution here is very elegant, but is also very unique.
Yes.
In the sense that 90% of the work of clustering is distance-based.
Yes.
And I'm not sure that introducing clustering via a very non-standard example would leave the right impression with the student. And similarly, the fact that everybody uses this EM example of multiple Gaussians is not only that people copy from each other,
of course.
It happens a lot. So again, this is the reason that this is that repeated example is that this is a very common situation that the students are likely to come across later on. So in terms of the educational content, I'm a bit worried about showing very elegant but somewhat non-standard, some would say esoteric application rather than the standard, maybe boring one, but the most useful.
Right. Right. Well, I can't help but agree to a point, but I do want to say, well, as for your first comment, the the underlying point here would be that, of course, 90% of the cases you just download a distance-based clustering tool and run it, and it produces good results. But the educational point here would be that still you need to understand your problem, and you need to look at it and actually see whether the standard methods actually apply. Because it is often the case that when you apply methods that are not supposed to work, they kind of work, and they produce some results, but the results are confusing and they are not good, and then you struggle with these results for a very long time. So basically the point here that I wanted to get across is that before applying the standard techniques, make sure that they are actually applicable.
Yeah. For a student who never saw any clustering, it would be better to see, it's an excellent example, but it's better to see it after they've seen and learned the standard distance-based clustering.
Perhaps, yes. Of course, I do not suppose that my chapter will be the only source on machine learning that a student will draw upon in his life. I mean, it's, yes.
One more question. So this is supposed to be for biologists. It feels like too much material for one chapter. And to really cover all of these concepts for biologists who are starting pretty far from this set of goals, I'm wondering what it is that they're really supposed to be able to do. And if they're supposed to be able to do these things, then the danger is there's so much material that you're going to have to bring in to really nail down an understanding of all these things. It feels like a lot of material.
Well, I don't really know how to answer, because it does not feel like a lot of material to me. It actually would feel like a very yeah, but I mean, I think it's not a good answer, but I think the previous talk would cover a lot more ground than this one.
I mean, this chapter will be good for undergrads in Sydney.
Yeah, sure.
Do you think that if you simply cut out EM algorithm, it's still within the chapter?
Sure, sure. It's not crucial for the logical flow. I mean, you can go straight from biprofiles to randomized clustering based on biprofiles. You don't lose anything. Yes.
There are so many topics in bioinformatics where EM algorithms come up more naturally.
Okay.
Okay, the last very short question. So I have one short question or comment or something. You have started with very simple and accurate, very nice formalization of the problem, and then you said that the problem will naturally decompose into two problems, and then proceeded with these two completely different algorithms. But I think in this scenario, most of the let's say machine learning textbooks will tell you, you should first investigate whether you can do some optimization on the whole model directly, because splitting the problem into two can lead into intact characteristics that you very two steps. So.
Yeah, I kind of skipped over. Well, it doesn't in this case. So yeah, obviously I will write about this, but yes, the problem can be safely decomposed in this case. Yes.